and visible?
Yes, yes.
Clearly?
Yes.
It's clear? Okay, how are you? All good?
Yes, sir.
Revision done? You are revising?
Yes, yes.
Okay, very good. Any doubts there? Anything you want to ask before we begin the class? Can you see the screen that I'm sharing?
Yes.
Yes. Okay, so let's continue with genetics and what we have to do today. Where were we? We discussed Mendel's laws, right? We started with the laws.
Yes, sir. We finished third law.
Okay, let me check. All codominance is done. Blood grouping. We also did some questions. Then second law, third law. Okay, we were on linkage and recombination, right?
Yes.
Okay, so give me a quick brief recap of the last class. Let's start from here. I'll help you, okay? So these recaps I ask you to do and students to do because they will put things in your head in a more better perspective. So when you revise your own study, it's good for you. I could have, like, if I give this give this recap, it will just be a recap. I know these things already. So tell me, let's start with you. We started with laws of inheritance, so let's begin.
So laws of inheritance given by Mendel is first one is law of dominance.
Yes.
Okay. It
As the word
that,
says. What does it say?
Okay.
Yeah, you can read. You can read on the board also. Just
Yes.
read.
In a heterozygous condition, one allele is dominant, expressed over the other allele for a particular character.
Yes.
Okay. Allele which remains hidden or is not dominating or not expressed is called recessive allele. Character, we took height, one character. Then there is a tall character and a dwarf character. Both comes in allele. These are known as alleles. Okay.
So they are versions, alternate
alleles
versions.
Alternating versions of genes. Okay. I think I have read somewhere like one more thing about them. They some related to coding something.
Code, yes. Yeah. They are like the sequence is slightly different, but majorly it's the consensus gene. Like the gene is same, but the sequence coding sequence is slightly different
in
for
the character. Both. Yeah. Okay. So we took idea. So if we take homozygous tall, homozygous means both what he can say, both
the alleles are same and dominant. Homozygous means same and tall is a dominant character, so both those alleles are capital T, capital T.
Okay.
Yeah.
If we take heterozygous tall, one is dominant, one is recessive. Okay.
Yes.
Then see, we took one more like
the
both
third
are
possibility.
Recessive. Yeah, both are recessive means dwarf. Okay,
yeah.
then we did crossbreeding between them. I mean monohybrid cross. Okay.
So this is just a monohybrid cross that we did. Yes. We started with a pure tall and a pure dwarf to begin with. Let's say the parent is tall and the other parent is dwarf. Then what happens? Yes, continue.
Okay. If one parent is tall and one parent is dwarf, then in the F1 progeny, we get the tall. What we can say? We get
all the progeny is hybrid tall.
Hybrid tall. Okay.
Yes. And then we do self cross.
Then we did self cross of F1 progeny. Then we got
yes. So why do we do self cross? To get the F2 generation, second generation.
Yes.
So self-crossing F1 progenies then tell us that 3 out of the 4 progenies were tall and 1 was dwarf. So the phenotypic ratio was 3:1.
3:1.
And tell me about the genotypic ratio.
And genotypic ratio is 1:2:1. Okay. Then
What are the 3 possible genotypes?
there are 3 possible. Actually, two or 3. I think 2. Tall and
3,
dwarf.
no?
Okay.
See here.
Tall. Okay.
1:2:1. You have 3 numbers. So 1 is. So you get all the possible combinations that we studied in the beginning. Do you know?
Yes. Yes.
Homozygous tall, heterozygous tall, and homozygous recessive dwarf. You get all of them. Yes, continue.
Then we did we got three types of visible characters. Or okay, then
3 types of characters.
No, then we did the first law given by Mendel, that is law of dominance. It states when 2 alternative forms of trait or character, we can say genes or alleles are present in an organism, only one factor expresses itself in the F1 progeny.
Yes, so this we have already done. No, this is the first law you talked about. Yeah. After that, we did the exceptions for the law of dominance.
There are 2 exceptions. One is incomplete dominance.
Yes.
In which like F1 hybridized exhibits character intermediate in the parent gene.
Yes.
Like so here like phenotypic ratio is not like same as the from the Mendel's monohybrid ratio. It is
so first tell me what do you see on the board? What is this?
Flower. Yeah, we did one example of flower color of snapdragon plant, that is Anthurium majus. The characters are 2, red, that is dominant, and white is recessive. Red is homozygous dominant, white is homozygous recessive. Then we did cross breeding between what is it? Cross. Okay.
We did not do anything. We have 3 possibilities: homozygous dominant, homozygous recessive. And in a heterozygous condition, instead of showing a dominant trait, it shows a blending. It shows intermediate trait, right?
Yes.
It should be showing red, but instead it is showing pink. Which means that there is an incomplete dominance in the flower color of snapdragon.
Yes.
Because there is blending. You understand?
Yes.
Yeah. This is the cross for that.
So.
And what happens to the genotypic and phenotypic ratio?
The genotypic ratio is 1:2:1, and phenotypic ratio is same, 1:2:1, and we are getting 3 types of
phenotypic ratio is not the same. Phenotypic ratio is changed. Phenotypic ratio earlier was 3:1.
Okay. Yeah.
Genotypic ratio stays the same.
But here it is same, like genotypic and phenotypic both are same ratios. So
I also told you incomplete dominant. Incomplete dominance, the phenotypic ratio is affected, but the genotypic ratio remains same. Okay. Right? Yes. Then we did codominance.
Now we did next that is codominance. The alleles which are able to express themselves independently, even when present together, are called codominant alleles. And this phenomenon is known as codominance.
So
we did one example.
So there is no recessive allele here, right?
Yes.
Yes. What was the example? Explain
The
the
example
example.
was our blood group, that is ABO blood grouping system. That is multi-allelic or polyallelic trait.
Yeah,
ABO
it's a multi-allelic gene. For
allelic.
one gene, there are 3 alleles. See, I've
Okay.
explained here alleles.
So ABO blood group is controlled by gene I, that is what you can say, gene I.
Yeah, the
Like,
gene is I,
yeah,
and it has 3 different alleles:
three
IA,
different alleles.
IB,
IB,
and small I,
IO. Yeah.
or small I.
Then
Yes.
then IA and IB produce RBC, we, like that surface antigens.
Yes.
Yeah.
So A group is because of A antigen present.
Yes.
Okay. And A antigen is a sugar. So IA gives rise to antigen A. So if someone's blood group is A, it has antigen A. If someone's blood group is B, it has antigen B. And if
And
someone's
I
blood
does
group
not
is
have
O,
any antigen. Yeah.
then it does not have any antigen.
Any antigen.
Now explain me this here, the genotypes.
Okay. In IA, you can say, yeah, IA and IB are dominant over like IO. So IA and IB are dominant alleles, and IO is recessive. Okay.
Yes.
And
For
then
A, just explain, for A blood
okay,
group, there are
for
2 possibilities.
A blood group, there are 2 possibilities: IA, and IA and IO. Okay, for B blood group, there are again 2 possibilities: IB and IBIO. And for O blood group, only one possibility is there, that both the genes should be recessive, IOI.
Yes.
Okay.
So where is codominance? I don't.
Yeah.
You only told that there is complete
When
dominance.
okay, when IA and IB are present together, both like express equally, and produce both produce, like we can say, antigen A and antigen B are present in the surface of RBC.
Yes.
So combine
Together. Okay, we did certain questions.
Yes.
Which, yes. Then we went to the second law.
Law of segregation.
What was it?
The law of segregation. According to this law, the 2 factors of each character assort or separate out independent of the factors of other character at the time of gamete formation, and get randomly rearranged in the offspring, producing both parental and new combination of characters. So it is a universal law, and it have no exceptions.
Yeah, in simple words, you can just say this: during gamete formation, each allele separates independently of the other allele in a gene.
There is no blending. There is no blending of alleles during gamete formation.
Yep.
So when 2 pairs of traits are combined in hybrid, segregation of one pair of character is independent of the other pair of characters. We can say like this.
No, that is for yes, one pair of allele trait.
Yeah.
For genes, it is law of independent assortment.
Okay.
So remember, I told you that law of segregation, the second law, and law of independent assortment, the third law. Basically, they are talking similar kind of things. About law of segregation is talking at the level of alleles within the same gene. Law of independent assortment is talking about genes in a dihybrid system, like 2 gene inheritance. So now here there are 2 genes here. Instead of 2 alleles in a gene, there are 2 genes which carry 2 alleles each. But basically, they are talking about segregation at the time of gamete formation only.
Okay. So law of independent assortment That what I told now. When two
Yes.
pairs of
So
traits
tell me
are
about
combined,
two gene inheritance.
two gene inheritance, we can say, if we take like two contrasting traits, that is one
Mhm.
height, and we took one seed. We can take one more one, and then there was there was like one yellow, a round yellow, and a wrinkled green. To another was there when I read the book. Okay.
Now look at the board
Yes.
here. There, okay.
So here we took two
We
traits,
took
height and seed shape, and
yes.
there were two traits. One was having tall, round, and another was having dwarf and wrinkled.
Yes.
Then from phenotypic ratio one, phenotype one, filial, we can say one. Okay, we got gametes, that is T, capital T and capital R, and small t, dwarf T and small t and small r. Okay.
Mhm.
Uh, when we when we combine them, we got hybrid tall and hybrid round.
In
Then
the F1. You have
F1
to tell this, yes, in the
in
F1.
the F1. Then when we self-crossed the F1, we got four gametes, that is T, capital T, capital R, capital T, small t, capital R, and small t, small r. Okay. Then we made okay.
We just
Uh,
cross them
we crossed them. Okay.
and
We
we made
got
a Punnett square.
points.
Now because there are four gametes in
Yeah.
each parent, so 4 into 4 is 16
16.
possible combinations are there. And then what happened? Explain me this side.
Then we got genotypic ratio 9:3:3:1, and here 9:1 we got the ratio same as the parental combination, that is tall r, tall t, tall r, and dwarf t, dwarf d, dwarf w.
Oh, you can simply say tall and round and dwarf and wrinkled. So tall and round and dwarf and wrinkled are parental combinations. They were present in the parents. But tall and wrinkled and dwarf and round are not present in any parent. So they are non-parental combination. Yes, continue.
Yes. Then we got phenotypic ratio, that is 1:2:1:2, 4:2:1:1. Okay.
So to remember this, the trick is it first increases 1, 2, 1, 2, goes to the
4:1.
maximum 4. You can remember it through the diagonal of that Punnett square. See 1, 2, 1, 2, then 4, then 2, 1, 2, 1. Okay.
Okay. Then total progenies has, you told, like 16. We got 16, 4 into 4, 16. And percentage of parental combination 62.5, percentage of non-parental combination 37.5. Okay. Happens due to the combination.
Yes.
Yeah.
Then
Linkage, what we did.
yes, we
Linkage
discussed
and
in the
combination.
last. Yes. Tell me about this, then we'll start.
So, yeah, linkage, when two genes having a physical association due to close proximity on the chromosome, which leads to a failure of segregation of two genes, that is linkage. They don't segregate or they don't separate. So therefore, the recombination is not happened. Okay. Then
Yes.
okay.
Okay. So from here, like I'll take it forward. So we had to start today about linkage, right? And then go to recombination. So write down write down linkage. Linkage between genes. Now, as I already told you, linkage basically is the physical association between two genes because they are present very close to each other on the chromosome, right? So if
Yes.
they are present very close to each other, the chances of them separating is very, very less, right? Now, how was this linkage found? So, and why Mendel could not find any linkage between any genes that he studied? So it was just sheer luck that the characters that Mendel chose were present either on different chromosomes, or if they were on the same chromosome, they were present at a distance from each other, right? So he could
Yes.
not see any linkage between any of the 7 characters and 14 contrasting traits that he studied. It was sheer luck. Now, there was another scientist who figured out and first gave this term linkage. His name was Thomas Hunt Morgan. Okay, T.H. Morgan also he's known as. But Thomas Hunt Morgan described linkage for the first time. Okay.
Oops.
Now, first obvious question is, how did he figure out linkage? Okay. He also coined the term. So the linkage term was given by him only. Okay. Now, how did he figure out linkage? So he was doing similar kind of crosses which Mendel did in pea plant. So Mendel took dihybrid or 2 genes and then saw that both these genes segregate independently of each other all the time. So T.H. Morgan thought that, okay, let me just do the same kind of process in some other organism. And he used the model organism. So he found linkage in Drosophila. You know what is a Drosophila?
So housefly?
No, it is a fruit fly. Housefly is different. Fruit fly are which, if you leave a fruit peel around or any vegetable that's ripened, then you will see very small flies hovering around it or sitting
Yes.
on it. They are different from the house flies. Okay, they have red eyes and brown body instead of a house fly who's like mostly black and gray, right?
Yes.
So they are very smaller as compared to house flies. So often people confuse them with the baby flies. They think that these are the babies of the house fly only, but that's not true. These are fruit flies, okay, a different species of flies. So these are very good model system to study genetics, okay? And Mendel found linkage in those. So what Mendel was trying to do is, write down Mendel's experiment, sorry, not Mendel, Morgan. Now we have to come out of Mendel. So what Morgan did, Morgan's experiment was, he was doing all different sorts of experiment. This is one of the experiment that where he found linkage, okay? So he crossed yellow bodied. So this is the character for the color of the body, right? Yellow bodied and white eyed. So he took a yellow body and white eyed female. Is it clear?
Yes.
And he crossed this fly, like he reproduced this fly with a brown bodied, which was another contrasting character. So either the body can be yellow or brown, and red eyed male, right? So you understand?
Yes.
One is for color of the eye, one is for the color of the body. And normally when you take any two traits, then what do you expect? You expect that when you cross it and when you reach the F2 generation, you will see all the permutation combination in the ratio of 9:3:3:1. Am I correct?
Yes.
Yeah. But he observed that in this cross, the genes or these two genes did not segregate independently. Okay. Now this was very interesting. And how did he figure out that they are not segregating independently? Through the F2 ratio. So he saw that in F2 ratio, in F2, which is generation, they did not follow 9:3:3:1, right? It should have been followed, right?
Yes.
10 should be parental, yellow body, white eye, and brown body, red eye. And then 3 plus 3, 6 should be yellow bodied and red eye, and brown bodied and white eye. This combination also should come up, right?
Yes.
But it was not in this ratio. And what does this mean? If it is not in this ratio, then the recombination is not happening, right? That's why we are not getting the non-parental types. So he found that yellow body, the gene for yellow body, is actually linked to the gene for white eye. And the gene for brown body is actually linked with the gene for. So basically the gene for body color, okay, was linked to the gene for eye color. Is it clear?
Yes, sir.
So whatever is the body color, if the body color is brown, then the eye color always most of the time was red. Okay.
Yes.
And this then proved that there is linkage between these two genes. If this was not followed, okay, they did not follow 9:3, which means now the percentage of recombinants is very low, which means there is linkage. So write down. Morgan stated that the higher proportion of parental gene combinations, was due to the physical association or linkage between the two genes. Okay.
Yes.
And he coined the term linkage. And both linkage and recombination, both terms are given by Morgan. He coined the term linkage to describe this physical association. And the term recombination to describe non-parental gene combinations. Okay.
Yes.
Okay. Now let's see how he got to know this gene. So you know that here, look at this example here. So if two genes are very close to each other, A and B, right?
Yeah.
The chances of segregation of them are very, very less during recombination. Because what happens during recombination is chromosome breaks and then rejoins. Okay.
Yes.
And this breaking and rejoining happens to non-sister chromatids. Now I will explain you recombination after linkage, but just know the genes that are farther away from each other have more chances of making non-parental combinations. Okay.
Okay.
Let's see how it works. So just give me 1 second. I will just can you see the screen now? No. You cannot see the screen, right?
Yes, sir. No.
Just one. think. Okay, let me first. Yes, can you see it back again?
Yes.
Zed, can you see it? Yeah. Okay. So before going to recombination, let's understand linkage we have understood. Now, before talking about recombination, I also want to tell you about a theory which is called chromosomal theory of inheritance. Okay. Have you read about chromosomal theory of inheritance?
Yes, two scientists have given that. Walter
Yes.
and Bovey.
Sutton and Boveri.
Sutton.
Okay. Yes. So this is important before going to recombination. So write down. Chromosomal theory of inheritance. Now, basically, when Mendel did all this work, okay, when Mendel wrote the book, did all these crosses, he worked for more than around 7 years on these crosses and doing all this study. But Mendel was not the person who actually gave the laws. Like, he wrote everything. The laws come from his work, no doubt. But it was not Mendel who actually wrote it in the form of laws. In fact, Mendel's work was not accepted.
Yeah. At the time they gave this law, he was not alive.
Yes, so his work was very, very underappreciated and unrecognized till the beginning of the 20th century. Okay, so he published his work. Before going into inheritance, write down certain things. So Mendel published his work in around 1865. Okay, and till 1900 it remained unrecognized. Okay.
Yes.
Now, can you think of why it remained unrecognized?
There were two, 3 things, and yeah.
No. So there were reasons for this. The first reason was that communication.
He was connecting between maths and biology, something like that. And
yes, great, you know it. So he was the first person who tried to explain biology using mathematical concepts, and this was something very, very new at that time. Okay, and so the biologists of that time, the contemporary scientists, did not accept it, did not accept the explanation or the logic, because it was very new and not very popular. People in that time were not very habituated to using mathematics to explain biology. Okay, biology was like living science, nothing to do with mathematics at that time. So first reason also before mathematics was that when he published, he could not reach out to many, a wider audience, or to a more number of biologists, because Mendel himself was not a biologist. He had his formal education in physics, and he liked mathematics a lot, and he was working at that time as a priest. Professionally, he was a priest in a church, and he did genetics. Interesting, no?
Yes.
He is like a very, very talented figure. Now, what happened was, first thing was lack of communication. Okay.
Okay.
So lack of communication, and because of lack of communication, his work could not be publicized. Write down, his work could not get publicized. So there was no publicity to his work. Many people did not know about it already. So it remained unrecognized. Second thing was, as you said, Mendel used concepts of mathematics, to explain biological process phenomena. Which was totally new, and unacceptable to many biologists of that time. Make sense? Zed?
Yes.
Yes, make sense?
Yes.
Third was that his concepts of genes. So he did not use the word genes. He did not know about genes at that time. So the knowledge about genes and chromosomes came later. Okay. So he used to call whatever is being inherited as factors. He said that this factor for tallness, this is the factor for dwarfness. He did not use the word genes or alleles. Okay. So third is, write down his concept of factors, in the bracket write down genes.
So he told like factors appear in pairs. Something.
Yes, his concepts of factors being stable and discrete. Units present in a pair. That never blend. Because Mendel did not know about incomplete dominance, he did not know about linkage. So about the exceptions, he knew nothing, right?
Yes.
So all he knew was that these factors never blend. So this was not accepted. So write down, they never blend, was not accepted by biologists, because they could see so much of variation in the nature. So actually what happened was, so one argument against Mendel was that Mendel said, if you cross a tall with a short plant, in the next generation all are tall, right?
Yes.
Then you cross a hybrid tall with a hybrid tall, in the next generation 3 are tall, 1 is short. So in Mendel's experiment, either the plant was tall or the plant was short. There was no plant which was in between, intermediate, right?
Yes.
Does it happens in humans also?
No.
No. In humans, we have humans of all possible heights, right? All variations are there. So every inch, every foot, you will find a human 5 foot 4 inch, 5 foot 5 inch, 6 foot, 6.5 inch, even to 7 feet, there are humans, right? So it's not like there are two categories, tall and short. So they also, if you see the human skin color, It's not like a flower color, right? Whether you will be dark or fair. There is a gradient, right?
Yes.
Some people are very dark, some people are less darker in shade for their skin, some people are fair, some people are very fair, right? So all these variations scientists could see in nature. And Mendel's concept of factors could not explain it properly because Mendel did not know about other things, other exceptions apart from what he studied in pea plant, right? So that's why his work was not widely accepted till 1900. Now, what happened in 1900? In 1900, there were three scientists. You would like to write down the name who rediscovered his work. So, first was Hugo de Vries. Second was Carl Correns. And third was von Schermack. Okay. These three scientists independently, separately and independently rediscovered Mendel's work. Write down. They rediscovered Mendel's work. And after that, the three laws were given from his work. Okay.
Yes.
The three laws that we studied above were given then. Is it clear?
Yes.
Mainly, Hugo de Vries had a big hand in giving the work the status of laws. Now coming to chromosomal theory of inheritance. So this was some history about Mendel and kind of work he did and why it could not get widely publicized and remain unrecognized. In 1900 only, around, like not exactly, around in 1902, like after 1900, 2 years after this, people started knowing about chromosomes, mitosis, meiosis, right? So this
Yes.
knowledge came up. And when this knowledge came up, then there were two scientists called Sutton and Boveri who studied chromosomes and how they are passed on from one generation to another. So Sutton and Boveri gave this chromosomal theory of inheritance. Okay. And what did this theory says? Write down. Chromosomal theory of inheritance states that the pairing or separation, the pairing and separation of a pair of chromosomes will lead to or would lead to the pairing or separation in other words, we call it segregation. Remember segregation of
Yes.
alleles and genes of a pair of factors that they carry.
So now also we are using factors, not genes.
Yeah, we use genes now. So factors are genes now, but at that time, when Mendel said, so this is just a validation. It is to validate that what Mendel did was indeed correct. And Mendel was correct when he said that, you know, there are factors which are present in pair, and if you have to make gametes, then they separate from each other, they segregate, one goes in one gamete, the other goes in the other gamete. He was right, because this can also be proven through chromosomes. So we have, for every chromosome, we have two pairs, right?
Yes.
One pair comes from
Father
Yes, and the other pair comes from mother. How many total chromosomes do we have in our cell?
In our cells, there are 23 plus 1 chromosome. That is
What?
like 24 chromosomes. Okay.
No, in our cells we have 46 chromosomes.
46,
23 pair.
20.
Okay. We have 23 pairs or 46 in a diploid cell. 23 chromosomes is present in gametes, sperms and ovum. So one copy, which is 23 chromosomes, is present in the sperm and the ovum. Okay. The other copy is present in the father, like in sperm, and the other is present in the ovum. When both of them fuse during fertilization, the first unit that we did, right, reproduction, then basically these two copies merge together, and we have, for every chromosome from 1 to 23, we have two copies, one pair.
Yes.
If you separate these, then the different alleles present on different chromosomes also get separated, right?
Yes.
Just like we were doing in Mendel's crosses, in dihybrid or monohybrid crosses. So this proves, so write down certain united. So this theory united the knowledge of chromosomal segregation theory united the knowledge of chromosomal segregation with Mendelian principles. Okay.
Yes.
Mendelian principles of inheritance. Is it clear?
Yes.
So both clear, chromosomal theory of inheritance and who raised to the status of laws and why the work was unrecognized.
Yes.
Yep. Please write this down.
So done.
Done?
Yes.
Okay, good. Now we can talk about what happens during linkage and recombination. Okay. Before that, I will tell you about certain things about chromosomes. So what are, there are terms like homologous chromosomes.
Yeah.
You know what are homologous chromosomes? Any idea?
Like during meiosis, they get separated and pass into different cells like that.
Yes, but what are they basically?
What are they?
They are the
homologous. Homo means same, logos, I don't know.
Yes, you're right. Homo means same.
Yeah. Locus.
So basically they are the same chromosomes, different copies of it, right?
Yes.
So write down about homologous chromosomes. These are two chromosomes which have the same length. Okay. Same short arm and long arm length. They have the same number of genes. Present at same locus on the chromosome. Okay.
Yes.
Now let me see if this works. Oh, it's very. So I'll making one chromosome here. Okay. This is one chromosome with Not right. Can you see now the screen?
Yes, yes.
Let's say this is one chromosome.
So we can take one red.
We can take one green. I'll use red for something else.
Okay.
And this is another chromosome. Now, basically, they are the same chromosomes. Okay, I'm just color coding it to tell you the difference. Now, in a chromosome, both the chromosomes, let me first mark things. So there is a short. So there is a short arm known as P arm. Okay.
Yes.
Can you see this is shorter than this in length?
Yes.
P arm, the word P comes from petite. You know what does petite means? Petite means tiny or
Tiny.
small. Okay. And we have a Q arm, which is the long arm. Together, this gives the total length of the chromosome. Okay.
Yes.
So this is basically the same chromosome. This is chromosome number 1. This is also chromosome number 1. As you said before, they separate during gamete formation. But one of this came from the mother and one of this came from the father in a diploid cell. You understand?
Yes.
Yeah. To begin with. Now on this chromosome, P arm and Q arm is clear to you?
Yes.
Now what happens is, just before division, this one is called chromatid. If it is a single thread, it is called chromatid. But just before division, it copies. So the arm gets copied. So it is just the copy of this arm. Okay. Do you understand?
Yes.
Just before cell division, this also makes a copy. So both the arms get copied. Now, this at the center is known as centromere. Okay. It's called centromere, but it's not all the time. It's not at the center, because if it was at the center, then both the arms would be equal, right?
Yes.
So it's not at the center. Q arm and P arm. Now remember, if it is on the same, if there are two chromatids on the same chromosome, these two chromatids which are present on the same chromosome, so they are exact copies of each other because they just got replicated from the same. So this is a chromatid, from the same chromatid two get made. So they are basically the same copies, photocopies, exact, right?
Yes.
So they are known as sister chromatids. What are they called?
Sister chromatids.
But these two chromosomes, because they are homologous chromosomes, both are chromosome number 1, but from different sources. So they are called homologous chromosomes. Now you understand what are homologous chromosomes?
Yes.
If one chromosome is chromosome number 1 and the other is chromosome number 3, let's say, then they will be heterologous chromosomes, different, because the genes will be different. On these chromosomes, the genes are exactly same. Do you understand?
Yes.
Yes. Everything is same. But what is the difference is that there can be a difference in the alleles. So let's say on this chromosome, can you see this red mark here?
Yes.
It is carrying the allele for the gene for tall height, and it is carrying the allele which is capital T, the dominant allele. Okay. Then the same allele will be present on this capital T. Okay, it cannot be different because they are sister chromatids. Same alleles are present on sister chromatids before recombination. Okay, but it is possible that on the other homologous chromosome, same gene is present, but it is carrying the recessive allele. That allele can be different, but then its sister chromatid will also have the same recessive allele. Do you understand?
Yes.
Yes. But do you see that the gene for height is present on both the chromosome at the same position?
Yeah.
This is called locus, position of the gene. Okay.
Yes.
And these two chromatids, if you take this chromatid and this chromatid, these two chromatids are called non-sister chromatids. Is it clear?
Yes.
Because they belong to two different chromosomes. Though both the chromosomes are homologous, but if they are copy from one chromosome, they are sister chromatids in the same house. And if they are from two different chromosomes, they are non-sister chromatids. Is it clear?
Yes.
Let's say there is another gene here close to this capital T, and this is for round. Okay, capital R. So this also will have round, capital R. But here, let's say from the other parent at the same locus, it is small r, the recessive one. And here it is small r, again, the recessive one. Okay, make sense?
Yes.
Are you with me? Yeah. Now let me take another color. I'll take purple. So can you see, let's say there is another gene which is here on the Q arm, the longer arm. And this is for, let's say, yellow and green seed color.
Okay.
Okay. So this will be capital Y. So the sister chromatid also will be capital Y. But on the homologous chromosome, let's say it is a recessive allele. So it will be small y.
Small.
And on the sister chromatid, it will be
small.
y. So do you understand?
Yes. Yes.
So this A, this is B, and this C. These are locus of genes, position of the genes. Is it clear?
Yes.
So this is how genes are present on a chromosome between two homologous chromosomes. Is this thing clear?
Yes, sir.
Anything that I'm leaving? Short down. Sister chromatid, non-sister chromatid, homologous chromosomes. Yes. Please draw this.
Sir, finished.
You done?
Yes, sir.
So if you are drawing with me only, pay
Yeah.
attention to understanding. If you need time to draw, I can give you time to draw later. Okay. So you understood everything
Yes. Yeah.
about the chromosomes?
Yes.
Just look at it. Take 2 minutes. Look at all of this, all the words, sister chromatid, homologous. Try to understand. Then we'll go to recombination. Okay. Make sure everything is clear in your head.
Yes.
I'll be back in 1 minute. Okay.
Okay.
Get some water. Okay, Zaid, I'm
Yes.
back.
Yes.
You're there. So everything is clear. Shall I move forward now?
Yes. Yes.
Now, during recombination, like during the process of meiosis, if you understood the chromosomes, what happens is there is an event that takes place, which is called crossing over
Yeah.
of homologous chromosomes. Okay.
Yes.
Now, in this event, let's see what happens. So let me again use the same color to make these chromosomes. Let's make, so you know now that this is the first chromosome. Okay.
Okay.
To the centromere. And then we have the homologous pair. Okay.
Yes.
This is the centromere. Now let's make the same genes that we took here. So what were the genes? Tell me. Had a gene for tallness carrying capital T. So this will also have capital T.
And small t, small t.
Small t and
Small.
then we had another gene close by. This was for, let's say, capital R. Here also it was capital R. On this chromosome, it will be small r and
Smaller.
smaller, right?
Yes.
Then we had a gene towards this, which is capital Y for yellow, capital Y for yellow. And on the homologous chromosome, we have small y for green and small y for green, right?
Yes.
These were the two homologous chromosomes we started with.
Yes.
Now, what happens in the first step of crossing over? One arm of one chromosome it actually physically crosses over with one arm of another chromosome. You understood?
Yes.
So this kind of arrangement is made. This is called crossing over. You understood?
Yes.
So the two legs here, you see the two, are they sister chromatids or non-sister chromatids?
Sister chromatids. Non-sister chromatids. Sorry.
Yes, because they are from two different chromosomes,
Yes.
right? So let's again mark the genes. Oh, sorry. Tell me. Capital T. Capital T. Small t.
Small t. And
capital R. Small r and small r. Then
capital Y, capital Y.
so here also it will be capital Y somewhere here, and this will have small y. So both of these capital Y, small y are now crossing over, right? And we have small y. So at this step, what happens is we call this step exchange. Exchange of
So can we say synapses?
Genetic material. Synapses is for neurons. Okay. We call it exchange. And this thing that you see here, this crossing over, this is called chiasmata. This structure where two chromosomes are actually crossing over with each other physically is called chiasmata, or crossing over bridge like formation. So this is like a bridge like thing. You understand? They both are crossing with each other.
Yes.
Chiasmata is clear? What is chiasmata?
Yes.
Now at this step, what happens is physically, from where they are crossing over, this arm breaks. So this green arm will break, this blue arm will also break. And in the third step, you will see that the first homologous chromosome will have one normal leg, and there will be a green associated with it. You understand?
Yes.
So that green part
Now
physically got
recombination is happening,
detached. Yes, physically got detached and joined to this blue region. And similarly, the other chromosome, with other homologous chromosome, we also have one normal arm, and at one arm that part will be attached. Is it clear? Zed? Is this clear? Now let's come to the alleles. So it was capital T, capital T. Keep telling me.
So capital T, small
Here.
t, small t.
Small t, small t. Okay.
Capital R, capital. Okay.
Then
Capital R, capital R.
okay.
Smaller, smaller. Then capital Y, small y.
Capital, you will see that here is a capital Y. What will happen here?
Small y.
Yes. And here? Here? Z.
Small y, capital Y. Small y. Sorry, capital Y, small y. Okay.
Capital Y and
Small y.
small y. Right? So
Yes.
now, can you see that in this third step here, basically what happens is these two are sister chromatids, these two are non-sister chromatids. So let's say there are 4 chromatids. The first one is this first chromatid. This is the second chromatid, the third chromatid, and fourth chromatid, right? 1 and 2 are sister, 3 and 4 are sister. 1 and 3 are non-sister, 2 and 4 are non-sister. Like this, right?
Yeah.
So tell me, what is the combination you have in this chromatid? First one. Capital T.
Capital T, capital R, capital Y.
Okay. The second one.
Capital T, capital R, small y.
First, tell me the fourth one.
Small t, small r, small y.
So these were the parents, right? To begin with, these are the parent chromosomes. So can you see these two chromosomes, which is chromosome number 1, sorry, chromatid 1 and chromatid 4, they are parental combinations, right?
Yes.
So we can also call it non-recombinants. Yes?
Yeah.
Now tell me for this second.
Capital R, capital Y, capital R, capital T, capital R, small y.
Yes, and here?
Small t, small r, capital Y.
So do you see that these two are different, right?
Yes.
these two are actually not present in any of the parent. They happen because of crossing over. So they are recombinants.
Yeah.
Okay, recombinants, or we can call it
Recombinant chromatids.
or non-parental types.
Okay.
Okay. So these two chromatids are non-recombinants, and these two chromatids are non-parental or recombinants. Is it clear?
Yes.
Okay. So just draw this clearly. This is what crossing over is. So see this, these two what do you say, gene combinations are not present in any of the parents, right?
Yes.
So do you now understand how do we get those 37% of the different combinations of genes?
Yes.
Here you can also see that the distance between T and R is less. You know, they are closer to each other. So in these two genes, there will be more linkage, right?
Yes.
So chances of they getting separated from each other is very less. To make them, these two genes separate and make a recombination with the other non-sister chromatid, you have to exactly cut it from the center, right? That very small space which is present in between, right?
Yes.
So the crossing over needs to happen here and it needs to get separated from here. But between R and Y, you see there is
very long space, long distance.
So there will be less linkage.
Yeah. Yes.
So if no linkage is there, then recombination will be more.
Yeah.
More chances of recombination can happen here. Okay, between the R and the Y. Make sense to you?
Yes.
These are the three genes that we have taken just to explain. Now, do you have NCERT?
Yes.
Open your NCERTs and come to page number 84. You cannot see the screen right now?
No.
Okay, just one moment. I will.
So that figure?
Yes, there is a figure on page number 84, right?
Yes. Yes.
So I'm just trying to pull it off for you, pull it out. Can you see the screen now? Again, this figure I was talking about. Can you see this figure, Zed?
Yes.
Okay. Now look here. Clearly, there are two crosses that they are giving you two examples. Okay, this is the first example, cross A and cross B. So first look at this one. They are separated from the center. Now look at this parental generation in cross A. The female, they have taken two genes. This is exactly what Mendel did. Okay, Morgan did. Okay, to find linkage. Do you see this Y and W?
Yes.
In your book, Y is for yellow body and W is for white eye. So in a female, can you see this Y and W together? Can you also see that the distance between these two is very less, right?
Yes.
Genes are very close. On the male chromosome, the Y plus, write down, the Y plus is for brown body. And W plus is for red eye. Okay. So the distance between these two genes are very, very less.
Yes.
So when you get F1 generation, in F1 generation, there is just the exchange of chromosomes, right? Without any crossing over. And when crossing over happens, you will see that 98% of the times they are parental type, which means non-recombinants. Yes.
Yes.
And only 1.3% are recombinants, which are non-parental type. Can you see here Y plus?
Yeah.
Y plus was for
Recombinants.
Y plus.
Y
Y
plus
plus was
for white type, that is male. Male
brown.
brown.
And W was for
Red. Male red eyes.
white. W
Yeah.
plus was for red.
Yes.
So this Y plus and W together is only coming 1.3%, which means there is very less recombination happening. So if in this case, in your first case, linkage is more, right?
Yes.
Due to which recombination is very, very less. So there is 98.7% of linkage. And that's why only 1.3% of recombination. You understand?
Yes.
Now let's come to the second cross here. They are taking now two different genes. One is W only, which is for white. Can you see white?
Yes.
The other one is M, which is for miniature wings, small wings. Okay.
Okay.
So you see that the distance between the color for I gene and the shape of the wings gene is more. It's more than what was it between the color for the body and color for the eye. Can you see it is more?
Yes. Yes.
So going through the same cross, now you see how many are parental types
62.8.
and how many are recombinants.
67.2. So there we did 62.5, 37.5, like that.
Yes. So what does it means?
Recombinants and parental type. No.
What recombinants and parental type?
Okay.
Recombinants are 37.2%, right?
Yeah.
It was 1.3% in this case. So first thing you can tell from here is that in this case, the linkage is
less.
low, right?
Yeah.
That's why there is
More recombination.
more recombination, right?
Yes.
Now, can you remember something with this percentage? This follows
Mendel's law.
Yes. And how can I say that this follows Mendel's law? Because in Mendel's law, in this F2, the ratio should be 9:3:3:1, right?
Yes.
And in this one, this 9 and this 1 are parental. And this 3 and this 3 is recombinant, right? What is the percentage of recombinants? You have already calculated this.
So 37.5. 37.5. I think so.
And what is the percentage of parental?
67.5. 62.5.
Does this percentage match with this percentage given here?
Yeah.
Yes. Yeah. So this means that the number of recombinants we are getting here are actually following law of Mendel. Yes.
Yes.
So cross B is following law of Mendel, but cross A is following linkage. Is having linkage. Is it clear? Just write this down. You can scribble with a pencil in your book if you have space. Don't use a pen, that will degrade your book, but write these things. And you have to write it on the figure only because this is what I have explained. So now can you understand the figure that's given in your NCERT?
Yes.
Let me know when you're done. Take your time, write down things, understand because these kind of things can be asked as a figure, or the same figure can be asked in boards.
So like case study?
I mean, you just explain the figure. What did you understand? So this is what I explained to you.
Okay, so finished. So I didn't drew the full image.
Yeah. So you can just scribble it on your book with a pencil if you have, where the figure is.
I will do it afterwards better. Is it okay?
Yeah. Okay, fine. But you understood, right? Is there any doubt in recombination and linkage?
No, sir.
Please ask. No doubt?
No.
So this was about Mendelian genetics. We did all the Mendelian genetics and we understood. Go through all of it once and let me know if you understood everything. Just go through all of your notes. Then I will start with the next thing. Take 2 minutes and go through all the notes that we have done till now, okay? And let me know when you're done. I'm here only. Okay, done, Zed?
Yes.
Any doubt?
Sir, not.
Please ask.
No, sir.
No doubt?
No, sir.
Okay, so I'm expecting that you understood everything, and now I can tell you about something different from Mendelian genetics, right?
Okay.
Okay, so after understanding all of this, let me tell you something about what Mendel did not know actually. So remember in the beginning, we talked about reasons that why Mendel's work was not appreciated or was remain unrecognized, because he could not explain things like human skin color, human height, and things through the pea plant experiment, right?
Yes.
Because these kind of inheritance are not studied by Mendel. So for example, one such thing is called polygenic inheritance. You have any idea about this term?
More than one trait?
Not more than one trait. It's just one trait.
Yeah, just one. More than, yeah.
What does that name suggest? Read the name again.
Poly means like more than 3, or like we can say what is trait controlled by 3 or more traits.
Where is trait in this word? Read this word again.
In polygenic, more than 3 genes. More than 3 genes.
Exactly. Simple. You are not reading the terms and you are thinking that you will understand. If you read biological terms enough, they give you the answer themselves.
Yes.
Polygenic, more than 3 genes control one trait. Okay.
Yes.
Write down polygenic inheritance. Write down those traits. Those traits which are controlled by those traits which are controlled by 3 or more genes. 3 or more genes along with the influence of environment. Are called polygenic traits. Is it clear?
Yes.
Till now, we have studied that Mendel talked about that one gene controls one trait. Yes?
Yes.
And one gene has only 2 alleles.
Yeah.
Capital T, small t, capital Y, small y,
Mhm.
round, wrinkled, green, yellow, purple, white, right? There was
Yes.
no third thing. And for and one gene only controlled one character.
Yeah.
T was not controlling color. T was controlling height only.
Right.
But there are cases where it's not true. For example, the example for this is what we discussed in the beginning. Write down. Human skin color and human height, for example. These are 2 characters that are controlled by polygenic. These are the 2 polygenic traits. So they are controlled by more than 3 or more than 3 genes. Okay.
Yes.
Let's take an example of human height. Let's assume that that 4 genes control human height. Sorry, human skin color. Okay.
Yes.
And these 4 genes are A, B, C, and D. These are the 4 genes. Okay. Now, you tell me what will be the darkest, and these genes, these 4 genes produce what gives color to the skin?
Melanin.
Yes. These genes produce melanin protein. Okay.
Yes.
So if we see, then this combination, which is homozygous dominant for all the genes at all the location, it is homozygous and dominant. Then for this, it will be the darkest skin or the lightest skin?
Darkest, maybe.
Why? What maybe?
All
Yeah.
are present.
All are dominant, all are homozygous, so all will produce lots of melanin. Simple. Yeah. So this will be the darkest shade. What will be the lightest shade?
All will be recessive, then they will be lightest. So small a, small b, small c, small d.
Right?
Yes.
Fairest or lightest shade of the skin, right? In between,
Yes.
there can be many permutation combinations.
Yeah.
Let's say if I make one recessive, if this gets recessive and this all of them remains dominant, then it is slightly darker. slightly fair. Then similarly, if actually all three are recessive but one is dominant, then it will be going towards darker. Do you understand?
Yes.
So different we see all different kinds of skin shades because skin as a character is controlled by multiple genes and their interactions. Also, so this is genetics, but also it is influenced by environment. Can you tell me how?
In the presence of sunlight, more sunlight, if there will be more sunlight, okay, we can take the regions of the earth, in the place of uh, if we take northern part of the earth, they are Polish area, I think. So their skin colors are white because of less melanin is produced, because less sunlight is there. Then if we take equatorial region of the earth, darker skin color is there, as more concentration of earth sunlight is sunlight is
Yes.
then
So more exposure to sunlight, darker the skin.
yes.
Less exposure to sunlight, fair the skin, because sunlight as a factor also controls. It is a environmental factor, right? Can you see this that I'm writing on the screen?
Yes.
That controls skin color, or in other terms I can say melanin production, right?
Yes.
Not this. Do you understand polygenic inheritance?
Yes.
Same is for height. You know, height also have multiple genes that control it. And can you tell me what environmental factors height depends on?
It depends on the altitude. Uh, uh, if a person is living on the equatorial region, he will be taller as compared to the that axial region of the earth, as there is
Where
more
is
gravity.
altitude then?
Sorry, gravity, based on gravity.
So person who are living on mountains, most of the time they are shorter.
Yes.
So those who live away from earth,
equatorial
tops, like towards mountain, not equatorial, not equator.
Yeah.
Equatorial is equator and polar.
Equator.
I'm talking about altitude. You said altitude, no? So altitude
Yeah.
is how you go up in the air from the ground. That is altitude.
Yes.
Altitude can increase on equator also. If there is a mountain on equator, you will be at a higher altitude on equator. You understand?
Yes.
Altitude means how far away you are from the center of the earth. You're right that equator is slightly bulged out, so it is slightly farther away from the center of the earth. But it increases also with the height, like on a mountain. So people who live in mountains are usually short statured, right? People who live in plains are usually taller, right?
Yes.
Okay. So, and also exercise, hanging, gymnastics, that also affects height, right?
Yes.
Along with the genetics. So this is polygenic inheritance. Noted?
Yes.
Done?
Yes. Yes.
Shall I move forward?
So mate, I have to write the last part.
Yes, please.
So finished.
Done?
Yes.
Okay. Second thing that I'm going to tell you is like opposite of polygenic inheritance. What was polygenic inheritance?
A trait is controlled by 3 or more genes.
Yes, one character, multiple genes. Now there is something called pleiotropic inheritance or pleiotropy. Pleiotropy is opposite. One gene which controls multiple characters. Okay, so write down. When a single gene can exhibit multiple phenotypic expressions. In other words, you can say when one gene controls many characters. Controls or influences, okay? Both. Then it is called pleiotropy. Okay?
Yes.
Now, an example of pleiotropy is in a diseased condition which is seen in humans. So there is a diseased condition known as phenylketonuria. You know about this disease condition?
Yes, sir. It is caused by the mutation.
Yes.
Or
Where?
In the gene coding.
Yeah. So this disease condition, which is called phenyl. You know why this is called phenylketonuria?
Because it reduces hair and skin pigmentation.
But what it has to do with phenyl or keto or nuria? The word has urea in it. Phenyl, ketone, urea. Have you studied ketone in chemistry? Aldehyde, ketone, carboxylic acid.
Yes, yes.
Have you studied phenyl?
Yes, sir. Phenyl. Yeah.
Have you studied urea?
Yeah.
So phenylketonuria in this disease, what happens is, you are right, there is one gene. A single gene is mutated. Okay. So let's try to understand. Single gene is mutated. And this gene it codes for an enzyme. So the gene gives rise to an enzyme. It is the enzyme ka gene. You know what is that enzyme called? Phenylalanine hydroxylase. Okay, this is the gene. Is it clear?
Yes.
Now this gene, normally when it gets mutated, what is the normal function of the gene? So this gene in the human cell, it converts, this is the normal function, converts phenylalanine. What is phenylalanine?
Organic compound.
It is an amino acid.
Okay.
Which gives rise to proteins, right? So it converts phenylalanine to tyrosine. Okay.
Yes.
Another amino acid. So this is what is the normal function of this enzyme is. This enzyme converts phenylalanine to tyrosine in normal cells. Now what happens if this enzyme becomes faulty? So if this enzyme becomes faulty, then this conversion will not happen, right?
Yes.
So the levels of phenylalanine will start rising up in the cell, right?
Yes.
In the body, it will start rising up. So when this happens, then what? It cannot get converted to tyrosine. So body automatically converts phenylalanine. Without this enzyme, the phenylalanine gets converted to phenyl pyruvic acid. Not a good thing, right?
Yes.
This thing is harmful. Okay?
Yes.
Harmful to the cell. So if phenylalanine starts increasing, then this will be formed, and this is harmful, so it causes effects. Okay, and what are the effects in this disease? First, it affects the nervous system or the brain, and it causes mental retardation. This is the first symptom. Now you see that this gene is mutated. So one gene, according to Mendel, if this gene was following one gene, one character rule, then it could have just mutating this gene would have caused just one effect in the body, right? Only one characteristic, any characteristic would have been affected. Yes or no?
Yes.
So if it is nervous system, then we will say that this gene is responsible for controlling normal brain activity. But it affects the nervous system. It also affects the skin cells, where it causes loss of pigmentation. Now, skin cells and brain cells are very, very different systems, right?
Yes.
Then how is one single gene affecting these two? Also, it affects the hair follicles. Now, this is another different system, very different from brain and skin. Here also, loss of pigmentation in the hair happens. Do you understand?
Yes.
So what do we learn from this? That there are various different effects. So this affects different characters. But in this disease, there was mutation in just one single gene. So this red pathway shows the defect. So this one gene affects multiple characters, then it is an example of pleiotropy. Is it clear?
Yes.
Please draw this chart. The normal and the affected both. Is this process completely clear to you, or is there any doubt?
Yes. Completely clear.
Very good. Because phenylketonuria will also come up again in genetic disorders. Okay, because it's a mutation in the gene. So if you understood it here, then it will save us some time there. Okay, so that's why I explained the whole disease here only.
So finished. Okay.